Hello, so today we're going to talk about nucleic acids. In this class, we're going to cover DNA and RNA. And the main objective is to examine the chemical structure of nucleic acids, DNA and RNA, and relate to their biological function in cells and in organisms. I'm first going to start with the center level, and then we cover the additional higher level of this topic. Okay, so why do we study this? Well, basically, we study the DNA mainly because it's the most important molecule on the planet. It's the most important molecule of life on Earth. All life on Earth, all cellular organisms are based on DNA. DNA is the code. DNA are the organisms. This is how we make our bodies. This is how we make our cells, our metabolism, and our traits. There are some exceptions. These exceptions are the viruses. Some viruses, they contain RNA as their genetic material. This RNA can be single-strand positive, single-strand negative, single-strand positive or negative, and it can also be double-strand. Okay, so some viruses have this exception. But away from viruses, all cellular organisms, all cells have DNA. And we're going to see that the DNA is comprised of nucleotides. We're going to examine each one of them and exactly how they work and what is the chemical composition and what are the bonds. We're going to see exactly each one of these three parts that make up them. Okay, so DNA is the genetic material of all living organisms. Yes, so DNA is the genetic material de facto of all cells, bacteria, archaea, and animals and all eukaryotes, because this was the genetic material of Luca, that is the last universal common ancestor. So Luca had DNA, we inherit this trait, we have DNA. All cells have it, no exception. The only exception, the viruses. So let's check the components of nucleotides. First, we have a phosphate group, here represented as this yellow circle. So this phosphate group has a phosphorus connected to 4 oxygens. Okay, then there is a link to a carbon and a link to a pentose sugar, here in the case of DNA, deoxyribose, DNA. Then in the carbon number 1, we have a bond to a nitrogenous base. This nitrogenous base are the letters of the DNA. This is what's going to be different from individual to individual, from population to population. And we have the 4 bases, A, T, C, and G. Okay, adenine, guanine, cytosine, and thymine. All right. So the carbon of the nitrogenous base is this carbon 1 prime. Okay, and the carbon of the phosphate is this 5 prime. We're going to see that these numbers, they actually matter when we talk about DNA replication and when we talk about polymerization of these monomers. Okay. So once again, we have phosphate, represented by the P, making a covalent bond with this carbon in the pentose sugar. This might be slightly different depending if you're talking about RNA or DNA. And then we have the nitrogenous base that is actually what differs from individual to individual. When we get the sugar phosphate backbone, that is the basic structure of DNA and RNA, we have phosphorus sugars, PS, phosphorus sugars, making this skeleton, this backbone. Okay, this doesn't change between individual to individual. And it's always 5 prime, carbon number 5 with the phosphate, with the 3 prime that is linked to the next phosphorus. So it's 5 prime to 3 prime end always. The bases, they're inside the molecule. They're forming these bases that are here protruding for the inside of the molecule. And they can differ. They can be very different depending on the organism. So again, we have another view, a single strand, only one strand, sugar phosphate backbone, and the nitrogenous bases here inside, A, C, T, and G. Once again, if we make a zoom, 5 prime end with the phosphate linked to the sugar, to the pentose sugar with the nitrogenous base, and then 5, 3, 5, 3, 5, 3, sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay, this is the backbone, DNA and RNA. If you have another single strand, depending on the composition, they can link, they can pair. Okay, how they pair? If they follow the rule of pairing, that are adenine with thymine and cytosine with guanine, A, T, C, and G. If you have the rule of pairing, if these bases are close to each other, they will attract each other and they will form hydrogen bonds. These hydrogen bonds are going to stabilize this molecule, these two strands, and eventually they are going to coil, forming the famous double helix that we're so used to see in diagrams and models on the internet. So again, the sugar phosphate backbone and the nucleotides inside. What matters for genetics? What is the genetic code? The sequence of pairing, the sequence of bases inside. Okay, so it can represent in this manner, as in this 2D view, or the simple pair, or coiling. Okay, compressing them and coiling, forming this very, very stable structure that is a double strand of DNA. It's actually very stable, way, way more than the RNA. It's very hard to work with RNA, but work with DNA sequence, manage, and manipulate is not that complicated. Once again, the nitrogenous base, we have guanine, adenine, cytosine, thymine, and uracil. Uracil is present in RNA, and thymine is not present in the RNA. Again, the complementary base pairing, this is in DNA and RNA, CG, and then T pairing with U or the adenine. Okay, we can see a pattern here. You can see a clear pattern. You're always going to pair one nucleotide, one base that has one ring with another base that has two rings. Why so? So then you keep the same length, the same volume in the double strand of the DNA. If you have two pyrimidines or two purines, for example, um, A, for example, A and C, okay, you're gonna change it. Or if you put like C and T, you're going to make a volume that is incorrect. Cells are going to detect, and we'll try to fix this mutation. When we sequence the DNA, and when actually we extract the DNA from organisms, the actual color is invisible. We don't see much. Okay, so these are samples of strawberry DNA, banana DNA, and human DNA. You can easily try to extract strawberry DNA at home. It's easy, it's straightforward, and it's cool. You're going to see that it looks like a cloud. It looks pretty much like a cloud of like water and gas, but basically it's all the DNA. As you connect a bunch of them together, they become this structure, but in fact they're invisible. Good. So we covered the basic structure of DNA, the double strand, and the nucleotides. So this is the structure, the photo that we're gonna see repeatedly over time. Once again, the rule of pairing, this is key. You need to memorize A to T, C to G. A, T, 2 hydrogen bonds, C to G, 3 hydrogen bonds. Once they stabilize, they coil, and they form a stable structure. The rule of orientation of always 5' end to 3' end. But when you pair two strands of DNA, they always have 5' to 3', but they're gonna flip. They flip. One here is 5 to 3, and another one's 5 to 3 here, and 3 to 3 there. So then they can make a opposite orientation, but a stable chemical. We have the purines that have two rings in their structure, and pyrimidines that have one ring in their structure. As I said, we always pair one purine with another pyrimidine, and so on and on. For example, here the adenine with 2 and the thymine with 1. Once again, guanine and cytosine. GC, 3 hydrogen bonds, A and T, 2 hydrogen bonds. Beautiful. And all this DNA, they make up long sequences. Okay, we're talking about something that is very, very long. Thousands, but commonly millions of base pairs connected to one another. So if you get your chromosome number 8, for example, you have two pairs, one from mother, one from father. Each one of these copies, it's a massive line of DNA with millions of base pairs connected to one another. And the genes, what actually matter, that is coding for a lot of traits and information and tools and enzymes in our body, they're inside these sequences. RNA is very different. RNA, typically, they have a single strand. They can form some loops, they can form pairings due to hydrogen bonds, but they're typically single strand. Okay? So what is the structure of RNA? Exactly as the DNA, single strand, covalent bonds, but you have a ribose. Okay, you don't have a deoxyribose. You have just a simple ribose here. And you have an uracil instead of U. This is what differs the RNA from DNA. The RNA can form a double helix depending on some structures, yeah, but it's more common to form loops and pairs, not actually form a stable double helix as the DNA. Okay, so just to recall, the DNA is deoxyribose, has thymine and double strand. RNA is a ribose, uses uracil, and is typically single strand. Types of RNA. Interesting. So we have some classes of RNA, and they are important for several regulatory processes and several biochemical processes in our body. The most famous one is the mRNA, messenger RNA, carrying the message to be translated, so then you can make the proteins. Okay. We have the tRNA, that is a transfer RNA. This is going to carry many amino acids during the translation process, so then you can build proteins in your body. So we can see that it's single strand, loop, some double strand, loop, loop, single strand. Then finally, we have the ribosomal RNA, very important, basically universal among all cells, and even the viruses, when they infect a cell, they need to parasite your ribosome to make their proteins. So the ribosomal RNA is a series of single-strand RNAs that match up. They interact with several proteins, and then you end up forming the ribosome. Okay. These are the most important ideas that we have so far. And the differences in the DNA, the letters of DNA, this is what's going to dictate individual to individual. Okay. The sequences of DNA, especially the ones that code for genes, they're going to dictate what differs species to species, individuals to individuals, and populations to populations. So all genetic diversity is based on DNA diversity. This is why it matters so much. And we also have the genetic code. You're going to study later this when we talk about mutations, when we talk about protein synthesis. But just to touch on this, the genetic code is a set of rules that biological organisms, even viruses, they use to produce the proteins. Okay, it's a series of triplets of codes, triplets of letters in the DNA and RNA that we code for a specific amino acid, one of the 20 amino acids. What matters for now is just to understand that all life is based on these codes. All life is based on DNA and viruses on RNA, and this is used to make the proteins. But don't mind so much about this now, because on protein synthesis, we're going to discuss each one of these codes and how they work. This is very interesting, and it's fantastic because it's universal. Even viruses use the exact same code that we do. One interesting activity that you can practice on your own to summarize this is drawing diagrams of DNA. This is an important skill, and it's useful for several IB papers, whether you creating the diagram, or you labeling, okay, the function, the part, the section, or the chemical. This is useful, and it's important for you to be familiar with, okay? Good. Here I simply took screenshots from Cognigy. If you have Cognigy, you can easily check this checklist, okay, of concepts or common verbs that you should be familiar with by the end of this topic, okay? Now let's go to DNA as a molecule of storage, okay? So the DNA, as I told you, is an incredible molecule in terms of processing and in terms of storing information, because you have the pairs, and you can have infinite possibilities with these four letters. How? Increasing the sequence. As you increase the length, the possibilities are endless. And this is what organisms do. They don't have a DNA of 4 or 10 length bases. No, they have massive molecules with thousands and millions of base pairs. Okay, so this is why the DNA is by far the best molecule that we have in terms of DNA, in terms of information storage. Taking, for example, a human, the amount of information, the amount of proteins that is needed to build a human, it's incredibly complex, and it's all stored on our nucleus and on our DNA. Okay, and all that just with 4 bases. But remember, 4 bases, and then you make pairs. With the pairs, you can have more sequences, and then you can extend the sequence. By extending the sequence, you can make up endless possibilities. Okay, for example, the human genome is around 3.5 billion base pairs, so it's massive, and it's divided by each one of these chromosomes. We have 23 chromosomes and the sex chromosomes. So each one of them typically rounds around some hundreds of millions of base pairs, starting from the biggest, number 1, and the smallest, 22. Okay, now talking about mutations and diversity. So we saw the rule of pairing. If you have a change in the pair, if you have a change in the letters that make up the DNA, this is what we call mutation. You're creating a new possibility. You're creating a new diversity. Usually, this diversity is bad for you. It's going to create a trouble. It's going to disrupt some system in the cell. But sometimes they might be beneficial. If they're beneficial, they're going to continue and they're going to follow and continue in the population. But most of the time, they're bad for you. They're problematic for the cell, and the cell tries to avoid as much as it can. There are several mechanisms to prevent mutations and to fix correct mutations once they were acquired. Okay, for example, if you take too much sun or if you ingest a certain chemical. And genome sizes are incredibly diverse. Okay, it varies greatly. There is a relationship with size and complexity, but it's not a straight linear relationship, but they vary a lot among viruses, among bacteria, among mammals, but especially if you compare like eukaryotes and prokaryotes. The numbers are pretty diverse in orders of scale. Okay. If we compare the human DNA and the chimpanzee DNA, we're going to see that they're very, very similar. Most of the parts we can match, we can compare, and if we sequence, we can clearly see similarities. Why? Because we share a more recent common ancestor with chimps than other species. But even like that, we're going to have several inversions and several mutations that happen during this time. But it's pretty easy to visualize similarities and match these blocks. Okay, now the HL part of this section. Okay, the first one is the directionality of DNA and RNA. First, we need to recognize the purines and pyrimidines, the role that they have as a pair. This we're familiar with. Okay, 2 rings or 1 ring, 2 rings, 1 ring. Backbone structure, sugar phosphate. But what happens? The direction of DNA replication is always 5' to 3'. But one strand is 5 to 3, the other one is 5 to 3 as well, but they are in opposite orientations. When you are in opposite orientations, you can better can better attract each other and stabilize all the molecule. And this is a joke that many biologists do. So what is the meaning of life? Is 5' end to 3' end. Okay? This DNA, once it's inside cells, especially eukaryotes, it's never free floating and open to the cell. No, it's highly condensed, it's highly compact because we have so much DNA. We have so much information that we need to organize in the best way possible. And we do this by linking to histones and nucleosomes. These proteins, they coil and they condense the DNA fiber. Finishing the additional higher level component of this topic, we have the Hershey-Chase and Chargaff's data. Okay, these are 2 concepts that we need to be familiar with before we finish. So the first one is the Hershey-Chase experiment. So back in the day, people were in doubt about what is the genetic material of organisms. Nobody was actually sure about it. So some group of people thought that maybe the DNA was the option. Some people thought about the proteins. So these 2 scientists, they came up with an incredible idea. Okay, so they took a virus, it's a bacteriophage, and they labeled the DNA with radioactive phosphorus, an isotope of phosphorus, and they made another group of viruses with the capsid, the coat, with an isotope of sulfur on their proteins. And they infected groups of bacteria. What is expected? So viruses, when they infect the cells, they place their genetic material inside, so then they can replicate and grow more and more. Okay, but what happened? What is the expectation? The substances that is radioactive, if it's inside the cell, they would be the genetic material. And if they are outside, they would not be the genetic material. So they infected both types, then they put in a centrifuge. So what they saw, that the ones that they were labeled with P, with phosphorus isotope, they were radioactive inside the pellet, inside the bacteria at the bottom of the tube. So which meant that actually the viruses placed this DNA with this isotope inside the cell, and they could see this in the bottom. And the experiment with the protein, they didn't see this. They didn't see the radioactive pellet, the radioactive bottom of the tube. And they observed a supernatant radioactivity. They observed a radioactive substance on the liquid surrounding the cells. So this was the final evidence, was a great evidence that actually the DNA is responsible for passing information from generation to generation over the course of life. Okay, not the protein. Proteins are essential, but they are the tools of life. They are what we use to make the processes. Regarding the Chargaff's data, it's a very simple concept that is basically a calculation. That is, in DNA, A plus G is always equal to C plus T. Okay, so let's suppose you have this chart here. Or if you have an organism, you're studying a specific animal, and some scientists tell you that you have on this animal 30% of adenine in its DNA. If you have 30% of adenine, you're going to have 30% of thymine. 30 to 30, you have 60%. Then the rest is 40%. If you have 40 divided by 2, you're going to have 20% of guanine and 20% of cytosine. This is Chargaff's data. Okay, it's a simple mathematical calculation that predicts the ratios of different nucleotides. That's it for today. Another interesting activity that you might do, you can find easily this on the internet, is this DNA origami. Okay, you can build, you can visualize the pairs, and it might be useful for you. Okay, thanks for watching, and I hope that was helpful.